everyone, how are you? My name is Marwa Bkerat, and just to give you a little bit about me, I'm really excited about this session today with Toddle. And this is my family. I like always to introduce myself by starting with my family. I'm a mother. I've been an educator for more than 20 years. I currently work at Queen Rania Teacher Academy in Jordan. I'm Jordanian. I've lived in different places. I lived in Saudi Arabia for quite a long time and then moved to Jordan in the past 4 years. I'm a fellow convener, an online workshop developer, and facilitator in MAPLI at Harvard Graduate School of Education. I'm an author for the book by Oxford called Biology for IB Diploma Program Course Preparation. I've been working for the IB for quite a long time, and I'm currently an IB examiner, IB workshop leader for different subjects and courses. And most importantly, I'm an IB fan. So this is just a little bit about me. Akta, you can carry on from there.
Thank you so much, Marwa. So in today's agenda, we will be exploring DP Biology, Investigating the Living World, assessment in the DP Biology course. Please bear in mind that this workshop has been produced independently of and not endorsed by the IB. It is not intended for it to be a replacement for any official IB workshops. Viewers and opinions expressed by the speaker are personal, based on experience, and should not be constructed as an official guidance by the IB. We have a few meeting housekeeping rules to go through for today. So this is a meeting. You can interact with Marwa. However, we request you to do so when we have sessions where we are having a pause and reflect, and the speaker is taking questions. You can put down your questions under the question tab. It should appear. While typing in your questions, do bear in mind that you are replying to all panelists and attendees, so that way we all can see your message and we can all interact with you. And I had mentioned this earlier, the webinar has been developed independently from and is not endorsed by the IB. And we also thank our speaker, Marwa, for generously sharing their learnings at this DP Thinkathon.
Thank you, Akta. Thank you.
Yes. And we have the last slide, which just says, let's get social. So if you would like to share with us your learnings from today, and we'd love to hear from you, you can do so by using the hashtag DP Thinkathon and tag Toddle across all social media handles. We have Twitter at Toddle_edu and Instagram at ToddleApp. Thank you so much. Over to you, Marwa.
Thank you, Akta. Thank you so much. So just to give you an idea about this workshop, so we're gonna just highlights on our learning objectives for the workshop. So today we're going to be enhancing your understanding of the purpose, structure, and requirements of assessment in DP biology. We understand that some of you could be new in IB or in DP, or others who could be expertise. So we'll try to cater the needs of everyone. We will share some classroom activities needed to develop student skills to enhance their achievement. And today I'm going to discuss with you some ideas of how to manage assessment in DP biology and overcome the challenges related to internal and external assessment. So this is in general about our learning objectives today. So to start with biology assessment, just to give you an overview about the DP biology assessment in specific. We know that biology is offered as higher level and standard level, and the main difference between the two is in the number of hours taught, in addition to the assessment papers. Yes, they all have the same number of papers, paper 1, paper 2, paper 3, and internal assessment. However, each paper has a different weight, could have a different weight, and could have a different duration from between HL and SL. So we can see that in paper 1, the duration is 45 minutes in SL, whereas in HL it's 1 hour and the same weight. In paper 2, there's quite a difference between SL and HL. SL is 1 hour 15 minutes and HL is 2 hours 15 minutes, with some difference in the weight. And paper 3, it's 1 hour in SL and 1 and 15 minutes in HL. However, internal assessment is the same for both categories, for both SL and HL. Okay. Okay, so we have a poll now. So I'm going to be sharing the poll. Please, you can scan this QR code or access the link in the chat that Akta will put right away in the chat and respond to that question. Which do you think is the most challenging in biology assessment? So we're going to wait a few seconds until you put your responses. Okay, so a few seconds to see the responses of everyone. Oh, that's interesting. I can see that 80% said internal assessment, and I totally agree with that. We have 22% dropping, 20% for paper 2. I agree with that, considering the database question paper 2. And in paper 1, we have 10% responded that paper 1, and paper 3, 10%. So from your responses, I can see that the majority of you highlighted that internal assessment could be the most challenging part of biology assessments. Some might consider paper 2 as the most challenging, as it includes some data analysis questions that could be a little bit challenging for students. Very few highlighted that paper 1 or paper 3 are challenging. Okay, since internal assessment, and I totally agree with you, when I taught biology for the first time, internal assessment was like the most challenging for me. But today we're going to try to highlight few things that will make internal assessment more, or we're not going to answer all the questions definitely, but we're going to be able to highlight few things that you do with your students in order to make internal assessment less challenging as much as possible. Okay, so we're gonna divide our session today into 2 parts. We're gonna highlight mainly the most of that session will go to the internal assessment, and we're gonna have some time also to discuss external assessment as well. Okay, so just few general guidelines about IA in biology. We all know that IA in biology is the same requirements at standard level and higher level. Doesn't matter if there's no difference between the two. We know that it's an individual piece of work. It is one scientific investigation that the student has to submit. It is made over 10 hours, and those 10 hours include the time for the teacher to explain to students the requirements of the IA, the class time for students to work on the IA and ask questions that could be face-to-face or could be through the class time itself. A time from consultation between the teacher and students could be one-to-one meeting with the student to highlight or to support the student in highlighting the research question. Time to review and monitor progress and to check authenticity of the work. So this is usually how we divide our 10 hours for the IA. And we know that the IA is 20% of the final assessment. It is assessed by the teacher and externally moderated by the IB. And we usually, the IB would request a sample to be submitted as part of the submission for the IB. However, we know that in the last 2 years, because of COVID-19, all the IAs have been submitted to the IB. So there was not a specific sample requested, but the IB requested for the submission of all students. And we know that it is assessed against 5 assessment criteria. So its mark is out of 24. However, it's 20%, yes, but it is out of 24. And it has to be 6 to 12 pages. Usually, we don't recommend that IAs get more than that. But usually investigations exceeding this length will be penalized in the communication criteria. Covers assessment objectives 1, 2, 3, and 4. And if we look at the IAs, it could be a hand-on investigation. It could be based on a spreadsheet for analysis and modeling. It could be based on extracting data from a database, from secondary data, for example. And it could be producing a hybrid of spreadsheet along with a hand-on investigation. So there's no one way of doing it. There are more than one way, and the student can choose the best approach that suits them. And it could be definitely used based on simulation or a comparison between a hand-on and a simulation. Okay, these are the grade criteria, and we can see that we have 5 grade criteria for the IAs. We have personal engagement, we have exploration, analysis, evaluation, and communication. And looking at exploration, analysis, and evaluation, we can see the bulk of the mark is here. Each of those is out of 6 marks. Personal engagement is out of 2, whereas communication is out of 4. Okay, so what we're going to do now, we have an activity. Akhta is going to break us into 5 groups now, and through the chat, a PDF of the IA criteria would be shared with you. If you don't have it, then you can access the document that is sent through the chat. Looking at this IA criteria, we now need to unpack these criteria and understand what is expected from students in each of these. So you will be divided into 5 groups. Each group will be working on 1 criterion. Group 1 will be working on personal engagement, Group 2 on exploration, 3 analysis, 4 evaluation, and 5 communication. Your role is to discuss what do you expect from students for each criterion. And you're going to be sharing your responses through the Padlet link that is provided here in the as a QR code here. You can scan it, or you can access the link through the chat, and that will take you to the Padlet where you can put your ideas and discussion and responses there. So I'm just going to share the Padlet here until is the group ready? Can we?
Ready, Marwa? Just give me 1 minute. I'm renaming the groups so that everybody knows.
Okay, until you do that, Akhta, I can share the Padlet link with everyone. So give me a minute for that. Okay, so this is what you will see when you go to the Padlet link. So each group will be working on 1 criterion. You will discuss the criteria based on the PDF provided. To make it easier for you, we've actually put the criteria in the chat. You can discuss it as a group and write your responses here of what do you expect or what do you think is expected from students in each of these criteria. Okay, so we'll just wait for the breakout rooms until they are ready. Okay, so we're back here. I'm going to share my Padlet again from here. Okay, we were looking at the criteria together. Okay. Okay, that's interesting. A lot of responses here. Okay. So looking at the responses we have, for example, let's start from the beginning with the personal engagement. Students show their unique ideas. Yes, with personal engagement, there should be something that shows that this is unique. It shows some input from the students. It shows some personal interest with the student. This is really important because personal engagement, it doesn't have to be as a heading, though I recommend that it should be something like under rationale or purpose or personal engagement. The students should write why they approach that question. And the question itself, it's not like a standard question, like the effect of changing temperature on rate of photosynthesis, where we know that we know the answer for that. So they should tweak it a little bit to make it unique, to make it to show some thinking behind it. With exploration, it's very interesting responses here that it has to include a very well-formed, focused research question, stating a hypothesis. By the way, a hypothesis it's not a mandatory thing to have under exploration. However, in biology, we do recommend that students write a hypothesis. It shows really good understanding and it supports them when they write their conclusion at the end. Explore ethical and environmental issues, looking at safety issues where possible. The research question must be focused, absolutely. Explore the methodology where the background information comes, the methodology used, presenting raw data, it comes in the analysis more, identify the material and apparatus used. Okay, and yes, in-text citation is required here, but we don't penalize it in this. If it's not there, it's penalized in the communication criterion. Analysis is where data should be accurate. Very important that students should refer not only to quantitative data, numbers, but also to qualitative data, which is basically the observations they see. Where relevant, some experiments might not have observations or qualitative data to refer to. And that is something many students would miss in their IAs. Having the raw data and processed data embedded within the IA itself is very important, because raw data here is part of the body of the IA. It's part of the analysis criteria. Statistics, how the data was processed, all the calculations, the graph comes in this criterion, and students need to choose the appropriate statistics, definitely, whether they're going to do average or standard deviation or t-test, ANOVA test, whatever the method they decide to do. Conclusion has to be very clear. It has to be related to the research question. Absolutely. I would say they always need to start with a conclusion statement, then justify this conclusion based on the values and data they obtained. And then they have to link it or relate it to theory where they compare it with other theory or other investigations that have been conducted. Communication, we look at how graphs and charts were labeled, x-axis, y-axis, titles and headings, units, if they're embedded within the graphs and charts, and tables. Decimal points must be, it doesn't have to be at 2 digits, but it has to be it has to be consistent, and they have to be related to the uncertainty itself. So if the uncertainty is 2 digits, then the numbers that come underneath has to be 2 digits. That is just one example. If the uncertainty is based on 1 digit, it has the numbers underneath has to be 1 digit. It should cover page and table. Definitely, they're not a must, unlike extended essay. However, because we only have 12 pages for the students to write, so usually 12 pages is not a lot. So we don't advise them to have a cover page and a table of contents if they're not able to keep the 12 pages. But if they are, then that would be fine. Okay, I'm going to come back to the raw data. The raw data is required to be as part of the IA. Some students would put the raw data as an appendix, which is not correct. Unlike extended essay, in extended essay, we only show, we don't have to show the raw data. We might show processed data, and that would be fine. However, this is not the case in IA. In IA, in biology IA, we should show the raw data completely. It should be embedded in the internal assessment itself. So going back to a few points, again, for personal engagement, it should show significant independent thinking, justification why they chose that research question. What is my personal interest in it? Does it have an impact on the community? Does it have an impact in the region I'm living in, on teenagers, on whatever? Where is the input, the personal initiative in that? For exploration, remember to have students identify a topic, identify a research question, and that research question has to be focused. Focused means they have to specify. If they're dealing with a bacteria, for example, they have to specify the name of bacteria. They have to specify if gender is something that they should keep. So it's done on gender, for example, on males aged between 16 to 18, just as an example. Background information is included here. Citation is important. Methodology is important here, and all the factors, all the variables needed. So they have to identify what is the independent variable. It's not enough to say the independent or dependent variable, but they have to say what is the value they considered for that. What did they do to change or to manipulate this independent variable? Similarly, dependent variable, what are they measuring? How they are measuring it? What instrument are they using to measure it? Control variables. What are the control variable? What is the value that it has been controlled at? And how was it controlled? What is the instrument? What is the device used to control it? When appropriate, they have to mention safety, ethical, and environmental issues. And this is very important. Like, for example, when they're dealing with humans as participants, a consent must be taken. Was a consent signed? Was the investigation discussed with the participants? Things to look at, like when they're dealing with bacteria, they should not conduct an experiment at human body temperature, which is 37 degrees Celsius. And that is according to the experimentation policy in IB. So a lot of things we can look at. For analysis, as we said, they need to have sufficient quantitative and qualitative raw data. It has to be there. Processing means all the calculations that take place, from the average to standard deviation to error bars to t-test or ANOVA test or whatever test they decide to conduct. Any graph that is processed here, it's also considered as part of the data processing, including the uncertainty. And as we said in biology, we include the uncertainty for dependent variables. If we are measuring, for example, mass, then if I'm using a balance that is made of one digit, then the uncertainty would be the least reading, which is plus minus 0.1. If we're measuring the number of bubbles produced, counting the number of bubbles, then the uncertainty would be plus minus 1 bubble, for example. Interpretation of data is very important. And when students interpret data, it's not enough that they mention what happened. The data supports my hypothesis. That is not enough. Or the data shows an increase or decrease. What type of data showed that? What are the values? They have to refer to the values. What is the quantitative data that supports this? What is the qualitative data? Did you have any outliers in your experiment? What do error bars tell us? Kind of variations from the error bars or standard deviation. Did I have enough sample size or not? So all of these things we have to consider in interpretation. Evaluation is basically divided into two parts. Evaluation criterion is conclusion and evaluation. And in conclusion, they must describe the conclusion as this increase, this will decrease, or justify this conclusion from the data obtained. This justifies, and based on the literature, so they're linking the values they got with the literature, with the justification for that conclusion, and they're comparing it to theories from before or investigations from before. And this is where we expect to see in-text citation. Okay, with regard to evaluation, it's not enough only to have positive weaknesses only. We need to have strength in addition to weaknesses, and if a weakness is identified, a suggestion should be a suggestion for improvement should be there. So these are the things we look at in evaluation. In communication, it's clear, the presentation is clear, well structured, necessary information on focus, the language used is important, it's precise, it's concise, subject-specific terminology used, proper citation, labeling of graphs, and use of decimal places, and so on. Okay, so I'm going to have a small activity with you just for 3 minutes. If you have a white paper in front of you, just a scratch paper, I'm going to ask you to draw a house, any house that crosses your mind, and you will be graded either 1, 2, 3, 4, 5, or 6. So any house that crosses your mind. So I'm going to give you 3 minutes for that. Any house that you can think of. Okay, we'll just wait for 1 minute and a half. Okay, and based on the house that you drew, I'm going to ask you to mark your house based on the criteria in front of you. So if does the house you drew has a door? If it has a door, you can move to the next category. It has a door in the middle, or it has a door in the middle and drawn using straight lines. So if you feel that your house is of a good example of that description, then give it the highest grade. So if your house is drawn in the middle with really straight lines, give it a 6. If your house has a door that is drawn in the middle with somehow straight lines, give it 5. We'll move to the next part. The house has no windows, or the house has windows, or the house has more than one squared window. 2D drawing, and if there is a garden or not. So I'm going to give you an example of this house. This is a house that is drawn, and we want to together to mark that house. Just to clarify a few things here. The numbers that we see here, this is called the mark band. The description here, the house has a door, the house has no windows, that's called a descriptor. And the level that you decide from the mark band, the 1 or 2, that is a level. So looking at that house, let's mark it together. The house has a door, yes, so I'll move to the next descriptor. The house has a door in the middle, that is right. The house has a door in the middle and drawn using straight lines. So I see that we're covering this category here. So I think the door is really drawn well, so I'm going to give it a mark 6. The house has no windows. No, my house has windows. The house has windows. The house has more than one squared window, but the windows are not straight and not really squared. So I'm going to give it a 5 here. I'm going to highlight that. Okay, the house is 2D drawing. Definitely that is not a 3D, and that is a good 2D drawing. So I would say 3. And there is no garden, absolutely no garden. So it's up to, and not even an attempt to have a garden. So I would consider it 1. So looking at the marks we have here, choosing the best one or the best mark is called best fit approach. So I'm going to look at the marks we have here. I have 1, I have 3, and I have 5 and 6. So it's not getting the average, but I would say that a fair mark here would be 4 out of 6. So this is called, what I've just done now is called the best fit approach. And the best fit approach is not getting the average, no, but it's very similar to the way we draw a best fit line in biology in a graph. It is looking at the numbers we've had and come up with a grade that is somewhere in between to balance the numbers that we've received. So this is just a small activity to help you understand how do we do the best fit approach. Another benefit, actually, another aim for that activity is, if I ask you to draw the house now, after looking at these criteria, I bet you're going to draw it really well and you're going to get a higher mark. So it is really important that we share the criteria with our students and to help them understand these criteria. It's not enough only to know the criteria, but to help them understand it so that they can really achieve well in exams, in the IAs. Okay, so just one application of the best fit approach. So you can see here that the teacher here highlighted the methodology in this area. The topic and question were given 5. The background information was given 5. The methodology was given 3. And the safety ethical issues was given 6. And overall, the teacher decided that the best mark for that student would be a 5, looking at the weight of the answers or the weight of each descriptor here. So this is just an example of how we do the best fit approach. So I would really advise you that you have the criteria for every single student, divide the descriptors in this way, and highlight the descriptors in each criterion and give a number there so that you're really accurate when giving the number. With time, after you practice this several times, you can actually do it without referring to or highlighting the criteria in this way. But I would really advise to do that. And I keep doing this no matter how many years I teach IB. Okay, these are examples of IA questions. And we said that the question should be focused. What does focused mean? You can see the independent variable here, which is in red, changing temperature. And we've specified the changed, the range of the independent variable. You can see that the dependent variable is there, pH of milk, which is measured by, and it's not enough to say pH, but how am I measuring it? So this is somehow a focused question that students can work with. Another example, is there a significant difference in the mass of leaves of the lemon tree? And note that for the lemon tree, we put the name, the scientific name of the tree, because the scientific name is required if you have an organism there. And which are present in shade compared with the ones present in light. So the independent variable is basically the place where the tree is growing, whether shade or light. And looking at this question, I would say a t-test would be a good statistical analysis here, because we are comparing two sets of data here to find if there's a significant difference or not between two things. So those are examples of IA questions that students can deal with.
Marwa, sorry to interrupt.
Yes.
We have a question in the chat box. The question is, do we start from the lower band or the higher band downwards? I think this was in reference.
That's a very good question. Actually, I would say start from the lower band going to the higher. We look at the higher when we want to know what is expected and how to give feedback to students. But we start from the lower band, moving to the following band in order to make sure that we covered everything required in each band and we're moving to the next. So I would say, yes, go from the lower to higher. Okay.
Sorry, we have time.
Yes.
There's just one more question.
Yes.
Does the 12-page limit exclude the appendix and reference? I'm not sure which page.
Yeah, we had that question actually in the past, long time ago. Usually it's excluding the appendices here, but I would say in IA, appendices are very few. We don't usually see them a lot because again, raw data has to be embedded within the IA itself. But if there is an appendix, it could be outside the 12 pages. Yes, that would be fine, I think. That's what we've received from when we were examining. Any further question?
Not for now, Marwa. Thank you.
Okay. Okay, so I'm going to ask you, what is your biggest challenge in the biology IA? And you can refer to the Mentimeter link there in the chat box, or you can scan that QR code. And to get your responses, I'm just going to go to the, okay. So we'd love to hear from you. What is your biggest challenge in the biology IA? Statistical test. I would say yes. It could be challenging, so it's worth, I would say you have to study, to read about statistical tests before actually guiding students and find a way of how to support them in statistics. As part of the introduction to the IA, you can actually go through some statistical analysis that they can conduct in IAs. You can help them to understand how to come up with standard deviation. What is a t-test? When to use t-test, when to use ANOVA test, when to use Pearson coefficient, for example. So that would be really good to review the statistics before students start conducting their IAs or doing the IA. Okay, time, I would say time, yes. Statistics again. Okay, I can see that most of you are asking about statistical tests, and I would say that introducing the IA, I would actually do something very similar with students. We go through the criteria, we take maybe 5 sessions to introduce the IAs, and as part of introducing the IA, I would take 1 session where I highlight some statistical analysis tips that could be helpful. I provide them with a document that could be helpful for them to share, to understand these statistical analysis tips, and highlight it as 1 or 2 sessions inside the classroom. It's really important for them to understand which statistical analysis to use. Okay, going back to, okay, I'm going to ask you another question, and we're going to come back also to answer some of your queries about IAs. What skills students need to develop to enhance their achievement in the biology IA? So I'm going to ask you to respond also to Mentimeter through that link that will be sent in the chat box, or scan the QR code. And I'm going to present your responses here. Okay. Okay, so what skills students need to develop to enhance their achievement in the biology IA? Analysis, absolutely. I would say that is the key, actually, because most students do not know what to do with data when they get them, do not know how to process these data, how to do, what kind of analysis to do. Citation in text or writing a bibliography, the art of questioning, very important. Reflection, very important. They need to reflect on what they have done. Definitely, they haven't done everything accurately, or we expect that there are some weaknesses in every single investigation. Conducting an experiment, absolutely. Research, how to research. And we keep telling them, you're not going to reinvent the wheel. You have to do some research to come up with your question, but you need to make a tweak. You need to have your idea, your thinking behind it. Okay, so just few tips to support students in IAs. I would say that timing of the IA is extremely important. I would say that many students, many schools I've seen, they do it by the end of grade 11. I would really recommend that. Some would do it beginning of grade 12, and that is fine, but timing the IA. There is some time where you discuss the question with the student one-to-one. You help them to narrow their ideas, and then they come up with a question, then they submit an outline for the IA, you approve it, and then they submit the draft and then the final. Practical skills, very important. What type of practical skills they need? Mathematical and analytical skills that we just mentioned. They need to learn how to do some statistics. Understanding the criteria. They need to understand it to be able to achieve well. IA overview for students. You must have some overview. I would do very similar thing like a workshop for the students to introduce them to the IA and what they can do to support their students, what they can do to understand about the criteria, to understand what is the expectation from them. Allow them to mark some samples, that would be fine. Looking at samples would help them actually to achieve more. Guidance and check-in sessions, very important that you have some hours with your students where you discuss the question. You discuss, even if they don't have a question, let them narrow their ideas. Okay, what topic do you like? You like plants. What are the things you can investigate about plants? Go search and come back to me, and so on. Standardization among teachers. If you have more than one teacher in the same school, it's very important that you work together. You have consistent, you are on the same page of your understanding about the criteria. You can swap your papers and do some standardization where you can discuss discrepancies in your marking. Feedback on draft is extremely important, not only as written feedback, but as verbal one as well, I would say. So these are some tips for internal assessment. After we can just take few questions if we have before moving to the external assessment.
We had one question that Arshia had asked before when you were doing the grade criteria. The question is, should raw data be at the material and analyze data inside result?
Okay, so raw data should be part of the IA. You can have, the students can have a heading called data, for example, raw data and processed data. They can have them as separate headings. It doesn't matter as long as they are part of the IA. They're not in the appendix. They are part of the IA. I'm not sure if I answer your question, Shahad.
Yeah, one more question again from Arshia is that some students put all the analysis inside the IA, for example, take snapshot of the Excel ANOVA and put inside results and discussions. Is this acceptable?
It depends, actually. They should not be doing, it doesn't penalize them if they put some, but if it takes a lot of pages, it might actually, because it might impact communication. They don't have to show the snap, the pictures of that or the images of their work, but they should mention how they carried on with their analysis. So they use the t-test using a spreadsheet or using Excel. So they're just mentioning the approach they used.
One more question is from Vara. How do the students justify the result of the t-test?
The result of the t-test, they have to mention, for example, if the t-test, if the p-value obtained is below 5%, so that shows significant or no significant difference. What does that indicate? That the null hypothesis is rejected or accepted. So they have to use these terminology, accept null hypothesis, reject alternative hypothesis, confidence level is high. So these terminology is very important to embed within their conclusion. Okay.
Yes.
Any
other questions we have so far? Yeah.
Okay, thank you, Akta. So I think how many minutes we have left? So probably very few minutes, right?
Yes, another half an hour.
Okay. Okay. So looking at the external assessment, the external assessment basically is basically made of paper 1, paper 2, and paper 3. And we know that for the current year, due to COVID-19 situation, paper 3 was canceled in grade 12. However, it is expected to be in grade 11 because those students currently in grade 11 will take their exams next year, where we expect the paper 3 to take place. The main difference in HL and SL that is the duration. So basically is in SL, it's 45 minutes, where HL is 1 hour. Because we have difference in time, so we expect different number of questions. So we have 30 multiple choice questions in SL, whereas we have 40 multiple choice questions in HL. The SL paper, it covers only core, where the HL paper, it covers core and higher level or additional higher level. The use of calculator, this is not permitted at all for SL and for HL. They don't have a reading time. We usually expect students to have 5 minutes reading time in all papers except for paper 1, which is the multiple choice paper. So in this paper, there is no reading time. And in this paper, there is no calculator allowed. And that is why some calculations like magnification, students need to carry on without a calculator, and usually those are not really difficult to process. They're usually simple numbers that they can work with. Something to keep in mind in paper 1. We know that there are some practicals which are obligatory. Some of these practicals, paper 1 might have questions related to some of these practicals. So it's very important that students study these practicals and refer to them when preparing themselves for paper 1. And by the way, paper 1 and paper 2 usually come on the same day of the examination. Paper 3 used to come on a different day, but not for the current year, absolutely. But usually it used to come on a different day, I mean. Okay. Any question about paper 1 before moving to paper 2?
We have a few questions from before. If I could take that. So Hind has asked, is it okay if the students used equipments for testing results from another lab, not only in school?
Yes, that is definitely fine. However, I would say that the school needs to request an evidence from that lab, like a letter or something indicating that it's the student's work. And I would ask the student to obtain photos evidencing that it's their work and not the lab technician at that lab.
Okay, we have a few questions coming in now for paper 1. Is it fine if students in grade 11 finish their IAS in the first year?
I would honestly say finish it in the first year. I would definitely recommend to finish it in the first year. The more burden you can take from students in grade 11, because in grade 12, they're really overwhelmed with the number of things they have to submit, like extended essay, TOK, orals, they come in the second year. So anything that you can remove from them and do it in year 1, go ahead and do it. And even if the student did not cover enough material for the IA in year 1, that is fine, because definitely they have to go and research and read about the topic they need to cover. So if they haven't covered human body, they can come up with an experiment related to human body. It's fine, as long as they research and come up with the information they need.
The next question is, do all the practicals in the guide, should it be done in the lab?
Okay, the practicals in the guide are not always practicals that they don't have to be conducted in the, if they don't have the material for that. But students need to be aware of it. They can show a video, they can do it virtually. They can have a virtual lab for that. Students need to understand that by conducting it in the lab, learning it from video, learning it from the lab, as long.
Marwa,
but
I think
definitely when
breaking.
Yes.
Your internet, I think the voice is breaking a little bit. If you could just repeat the answer again, that would be great.
Yes, I noticed that. Okay, again, I was saying that it doesn't matter if they actually do the experiments. They can do it in year 1, as we said. So the last question was not clear, Aktha. This is the one I broke at.
Yes.
Can you hear me?
One. Yes, we can hear you now.
Yes, can you repeat that?
Do all the practicals in the guide have to be done in the lab?
Again, we said no, they need to be, it could be done through a video, could be done through virtual lab, could be done through hand-on experiment, as long as they understand what is expected in that practical.
We have 2 more questions. Would you want to take them now? Okay. How many MCQ questions from paper 1 should be similar for HL and SL?
It differs actually from one year to another. They're not always similar. Sometimes they have similar questions. So if you look at 2 papers that were given at the same time, you can see some similar questions in both. But I cannot give you a percentage because that differs from one year to another.
The last question is, what could be the plan for IA if we have limited tools in the lab?
Okay, they can do a spread. They can have their IAs based on secondary data, where they get the data from a spreadsheet, a database, and these data, they actually deal with it and do the analysis for it. That is one option. So if they don't have enough material, or if they don't have, if they did not get the chance to conduct an experiment, they can do that. Another example, they can actually work with plants, because most experiments with plants do not really require a lot of material in the lab. They can work with plants, They can conduct experiments with plants that would be really easy and very simple to do. And they really give good results as well. Okay, moving on. Thank you, Akta. We can take more questions after we finished with external assessment. Looking at paper 2. Paper 2, some of you actually highlighted that paper 2 could be challenging at the beginning of this session. And I agree with you. I would say that paper 2, because it includes a long data analysis or database question, that could be challenging a little bit to students. And looking at paper 2, as we said, it differs between SL and HL. It's 115 minutes in SL and 215 minutes in HL. The weighting is basically different, and we can look at the mark because the number of questions is more in higher level. The structure of the paper is divided into section A, where you have a database questions, short answer questions, and then section B, where you have 1 out of 2 for SL and 2 out of 3 for HL. And definitely they can use a calculator here. I would actually really recommend here that you have students practice database questions from day 1. The more they practice database questions, the more they become familiar with these type of questions, and the more they feel themselves confident in answering such questions. I would always tell my students, the database question is like the alien question. You don't need always the knowledge you have, but it's based on how you interpret graphs, how you interpret data, looking at the sample in that data, looking at error bars. If they give you error bars, they definitely mean something here. Looking at all these data together and what can you interpret from it. So it's basically skills in interpreting data. And the more they do, the more they practice, the more they become able to answer such questions. The extended response questions in section B, I would highly recommend that students actually answer them in the form of bullet points, not essay writing. I always believe that bullet points, its structure and structuring the question, it gives a structure to the answer. It helps students organize their ideas, their thinking, putting point at a time, 1 point at a time. If the question is drawing, then definitely they have to draw with pencil, no use for colors because colors do not show in papers. They have to use really clear drawings. They have to have at least their drawing as half a page, not too small, so that all the details are clear and labeled properly. Any question about paper 2?
Yes, we have a few. Any sources from where we can make students practice the data questions?
Okay, the IB Bank question, they have a lot of database questions. You can refer to past papers. Every year in every paper, there is a database question. And you can use these questions whenever you cover a topic. Look for the database questions related to that topic and introduce it to students and have them practice those questions.
The next question we have is, what do you mean by practicing database questions from day 1? How do we do that?
Okay, that's a very good question. I mean that, let's say that you started with cells, for example, with the first unit, and you covered, I think there are no database questions related to the first topics, but there is one related to facilitated diffusion. That is the first that you can actually, and it's a simple one, you can use that one. And for every unit that you cover, when you finish the unit, you finished unit 1, for example, follow it by practicing some database questions related to unit 1, and do the same with unit 2, and do the same with unit 3. You can solve questions with them in class. You can give some questions as homeworks, but I would say always go back to solving these questions and help students understand from the mark scheme how to solve these questions.
The last one we have is it true that they need to answer in the box only?
Yes, it is true, because anything that is outside the box and as an examiner, I might not be able to see it. So we always advise them to answer inside the box because the examiner only would see the box. So anything outside, we don't see it. So if they need to have more space, they can write, continue on answer sheet, and they have actually an answer booklet that is accompanied with the exam. So they can put their answer in that booklet and highlight that this is the continuation of question 3, part A, for example.
We have one more question in. Should they draw using a pen or pencil?
Definitely pencil. Labeling could be with a pen, but the drawing should be with a pencil. Let them avoid using arrows to label because the point of the arrow where it's pointing towards the part they want to label sometimes is not accurate. So make them use straight lines. The straight line should be used done with a ruler. Do not label things inside the drawing. Have the labels always outside the drawing to make it clear and easy for whoever is the examiner looking at that paper. Okay, I'm going to move to paper 3, and then we can take more questions if you want. So in paper 3, the duration of that paper is 1 hour for SL, it's 1 hour 15 minutes for HL, and we can see that there's a difference in the number of questions as well, and the mark. Many students in the past, in paper 3, they forget section A because section A is related to the experimental skills, to the practicals. So that part, everyone has to answer it. Section B is the part where they select the option they want to answer. And it's very important that students know which option they covered so that they select the option they want to respond to. So in section A, and both sections are obligatory. However, in section B, they choose the option. I would say paper 3 is sometimes challenging in the type of database question because the data analysis question that comes here is a little bit different from that in paper 2. It's more related to a practical skill or an experimental skill or a technique. But again, make sure that students practice these papers, practice paper 3 and get more familiar with it. And again, the calculator here is permitted. Any question about paper 3?
So far, there's nothing in the chat box, but I remember in the beginning, somebody did ask that, are they doing away with paper 3?
Are they doing again?
Are they doing away? Are they getting rid of paper 3 anytime soon? There was a question.
Yes. No, it's not. It's only for current grade 12 students. Paper 3 is not considered due to COVID-19 pandemic. So that is a temporary situation for current grade 12 students. However, for grade 11, current grade 11, as far as we know that paper 3 is still included for the current grade 11 students. We don't know yet if it has been canceled for them or not, but I think that would be clarified by the end of this year. But for the current grade 12, no, yes, definitely paper 3 and group 4 project is canceled for them.
We have a question that says, how do we plan for paper 3 during the session?
Okay, I would say paper 3 is not, usually we have to help students look at these practicals, understand the practical. You can have them, when they finish learning about the practicals related to a specific unit, you can give them those questions that came through over the past years in paper 3 that are related to that unit in specific, and have them cover answer questions related to that, because this is a little bit challenging. They need to look at these practicals, understand the practical very well, and understand how to respond to such questions. So again, practice more papers of paper 3. With regard to the option, they need to study the option, and once they finish the option, have them practice paper 3 for that option from the past papers that have been done in the past. Okay, now this is an activity where I want you to look at the link that you have here, and Akhta will put that link in the chat box. And let's respond to that question. How can we help students achieve higher results in the biology examination paper? So I'm going to share that from my side here. Okay. Let me.
Marwa, you're breaking up a little bit.
Okay, so I'm sharing the Jamboard. If you can share that in the box.
It's done. It's in the chat box.
Okay, so again, how can we help students achieve higher results in biology examination paper? So we would like to hear from you. What do you think we should be doing in order to help students achieve higher results in biology examination? Okay, I can see some answers here. Constant review and reinforcement of learning. Absolutely. The more we review questions with them, review past papers, definitely practice. And I would say practice and practice and practice. Whenever you finish a unit, practice questions related to it. Maybe it's not wise to give them a whole examination paper at that time, but give them questions from the IB bank related to that topic. And you can filter the questions there just to include topics, questions related to specific topic. Encourage them to practice more questions. Give them more practice questions from the IB bank and past paper. Encourage them to read and research, absolutely. Okay, so we're waiting for more answers to come. Give them more practice, especially in extended response questions. I will definitely say that. Okay, so practice questions to do peer evaluation as trials. I like that. That's very good when they mark questions, and always mark like an IB examiner. Be mark based on the mark scheme. Consider the mark scheme when you give them questions to practice. Use games to review. That's very nice. Kahoot, by the way, could be used for the multiple choice questions. Very interesting. You can give them different games to review questions related to data analysis and so on. Okay, a few tips from my side. I would say they're really important to understand the command term. What does it mean to say compare or compare and contrast? So I would always say for students, command term, you don't teach the command term as a standalone, but you teach it as you move on in your curriculum, in your syllabus. So when we talk about cells and the different type of cells, eukaryotes and prokaryotes, we highlight distinguish. What does distinguish mean? Put differences in a table. Make sure that you have one point and parallel point. Compare means similarities. Compare and contrast means similarities and differences. So very important that they understand the command terms. I would always tell them outline the answer before you answer it. And that is particularly for extended response questions. So you would see that discuss what are the enzymes in DNA or the role of enzymes in DNA replication, for example. I would tell them, highlight the keywords here, enzymes, DNA replication. Outline your answer before you answer it. Okay, what are the enzymes? Okay, I need to consider polymerase, DNA polymerase, 3, 1, ligase, helicase, and all the enzymes related. They can just write them before they answer. DNA replication, what do I need to look at? Leading strand, lagging strand, Okazaki fragments. So they're just outlining their answer before structuring their answer. The outline at the beginning would really help them to structure their answers and not to forget any points, just to summarize everything at the beginning and then start to structure their answers. Provide answers in bullet points. I would recommend that a lot, particularly with extended response questions. It gives them a way to organize their ideas. Highlight keywords and questions, as we said. Drawing must be clear and properly labeled, and most important thing, help them to solve past papers. The more past papers they solve, the more they get confident and comfortable with the questions. And believe me, many questions over the past years have been repeated, but tweaked a little bit or changed a little bit, but it always refers to the same question. Okay, so we can take further questions from you if you have. If you can.
I think we have a few. How to manage teaching all options if students opt for various options?
Okay, I can't see that. How to manage? Can you repeat the question? Because I don't see it.
How to manage teaching all options if a student opts for a lot of options?
Okay, we know that we have 4 options and you can only choose one. I would say don't teach all options. You just choose the option you want to teach and you feel that it is suitable for your students, because the option that you choose will definitely relate to the equipment you have in the school. You can make the decision as a teacher what option to tackle, and that option would be taught to all, and that is the option you teach in class, and that is the option students can respond to. I would give you an example, like when I used to live in Saudi Arabia, doing ecology experiments was really difficult at that time. That was a long time ago. So I was unable to choose the option that is related to ecology because I could not have ecology experiments related. However, the option I feel comfortable with is like option D, human body, and that is suitable to everyone. So again, you can decide the question as the option as a teacher. Many schools would give the right to their students to decide it, but the only problem, maybe you can vote and have students decide on one option.
The other question we have is, when do you teach the option, first or the second year?
Okay, the option is I don't teach standalone. I don't teach the core or higher level or option as a standalone. We integrate the topics. So for example, let me give you an example. So for example, you're teaching photosynthesis in core. There is a lesson in higher level that is also called photosynthesis. So I would teach photosynthesis in core, then higher level, I would teach photosynthesis higher level. Now, with regard to the option, I do the same, but because the option I teach is human body, and human body option, it could be integrated with topic 6 in core and topic 11 in higher level. So I would teach, for example, digestion in core, cover digestion or related topics in higher level, and do nutrition in option, for example. So we always integrate the topics, and I usually teach human body as a whole big unit in the second year of the program. But again, you teach it whenever you want. Some teachers prefer to do it in the first year. Again, it's up to you.
Can the option chosen change each year?
No, because the option you choose, if you're talking about the same group of students, so if you choose an option in grade 11, that has to carry on with them to grade 12. But you can choose a different option for the following grade 11 students. So for each group of students, you can choose different option if they are in different years, one in grade 11 and one in grade 12. But I would say that would be really sometimes difficult for you, as you prepared all the content and preparation is related to one option. So again, it's up to you to decide.
Those are all the questions we have, Marwa.
Can I ask a question?
Yes.
Of course, yes.
Yes. Please, can you tell us more about GenBank and GMOL?
About, again? Mona, I think I'm losing you.
Can you tell us more? Yes. I'm back again. Can you tell us more about GenBank, the one for the sequence, and the data bank for the genes?
Okay, I'm not sure what
just a small description.
Are you referring to the IB bank?
No, no. Some of the experiment on the IB book, I think it's Hodder maybe,
Yes.
refers to a gen bank where it's a data bank for genes and for sequences. So some of the experiment or the application might be taken from there. And it's the gene bank, okay. But it's there, its name, like the name of the site is Gen Bank. It's not Gene Bank, okay.
Okay, I'm not sure honestly, but yes, I remember there. I'm not sure if the name is the same, but there is something simulation that you can use for karyotypes and genes. But I'm not sure about the bank itself, honestly, Muna. I think the idea is for practically you can use that one, or you can refer to others. That would be fine. As long as you use a simulation, you use something that relates as practical to that topic.
Thank you.
Thank you. Thank you.
I think with that, we've come to the end of the session, Marwa. So thank you so much for a wonderful session today. I'm sure everyone who joined in is taking away very useful insights. We'd also like to invite everybody to the Group 4 panel today at 11:30 a.m. GMT to learn more about supporting students with Group 4 projects. You can register through the IB DP Thinkathon website, or you can use the link that I'm dropping right now in the chat box to join us there. So that's the Zoom link that I have pasted in the chat box. And if you're interested in knowing more about how you can use Toddle in your school, we're also doing a quick walkthrough to show you what Toddle has to offer DP schools today at 10:45 GMT. The link for that is also being dropped in the chat box right now.
All right, Marwa, thank you so much. And you're so resourceful. And I have a question. Recently, in one of the IB groups, we have seen the latest first assessment in 2015. We have gotten a new kind of integration. Did you come across with that? Because I have that. If you want, I will share it with you.
Okay. Okay. I'm not sure if I got the question correct, but the curriculum for biology changed in 2016. So that is the IB question bank, or the IB bank is related to the new changes. So 2016 is the first exam according that came actually for the new syllabus at that time. I'm not sure if I'm answering the question correctly because you were disconnecting a little bit.
Yeah, Marwa, no, I'm talking about the new curriculum from 2023. They're changing.
Okay.
Yeah. So,
yes, it is changing.
And we are not going to have IAs anymore, and paper 3 is also not on the plate. So, do you have any idea on that?
Yes, yeah, I understand your points. Yes, yeah. Yeah, actually, there is a change in 2023 that is coming, and that would be applicable to not to the grade 11, to the current grade 11, or to the current grade 12. That would be applicable to the students who will be in grade 11 in the following years, not next year, the year after. So basically, there are many changes happening, but I cannot refer to that as these changes will be out by the IB. However, there is a document called Curriculum Review meetings, which highlights the discussion that took place in these committees and the changes that are expected to take place. And these are not the final changes, but these are the expected to take place. And definitely the structure of the papers differ a little bit, and even the structure of the IA would differ a little bit. And paper 3, it has been removed, but it has been, as you can see, it has been integrated in paper 1 and 2 in a way or another.
Thank you. That's all. Thank you. Thank you so much.
You're welcome. You're welcome.
That's all we have time for today. Thank you so much for being a part of the 300 educators who joined Toddle's first ever IB DP Thinkathon. All videos will be available on our website. We can't wait to share what we have planned for the DP, and we would love for you to partner with us to create a platform. So thank you, everybody. Have a good evening or the rest of your Sunday. Take care. Thank you,
Thank
Marwa.
you. Thank you, everyone.
Bye
Have a
bye.
nice
Thank you
day.
so much.
Bye-bye. Bye-bye. Thank you.